I'm here to present Spago Nanomedical in a nutshell, of course, since we have 10 minutes. Spago has developed a nanomedical platform which we can use to tailor nanoparticles to be used as either diagnostics or therapeutics. And we work with life-threatening diseases. We have chosen to start to focus on cancer, and I will get into our projects a little bit more as we go. First of all, just to say something about the technology that we have invented. We are tailoring a polymeric type of nanoparticles in a way so that they can accumulate passively in solid tumors. And the good thing about these particles is that we can tailor their size, which is critical to get enough material into the tumor. And they are also excellent chelators of metal ions, which means that we can bind metal ions that are useful, for instance, for MRI, which is our first project SpagoPix is dedicated towards, but also radioisotopes, so that we can use the particles for radionuclide therapy of solid tumors. We believe this is a unique platform. We have solid IP of this, and we intend to become the world leaders in the field of passive targeting to tumors. So starting with MRI, why is there a need for new MRI contrast agents, you may ask? This field is quite old. The agents that has been around for 25, 30 years now. But they're all rather unspecific. They are based on small molecules, and they are based on a metal called gadolinium. The unspecificity of these agents results in a very high percentage of false diagnosis. You see a lot of things in these images, especially when you're looking for tumors, and you don't really know what you're seeing. So this is a problem with today's use of MRI in solid tumors. The second problem is related to gadolinium, which is a metal that is not supposed to be in our bodies at all. And it accumulates in the brain, for instance. There's a huge debate going on in the field what this means clinically. But what it has meant or do mean already is that patients are getting worried, physicians are getting worried, and authorities are getting worried about this. There's also the fact that these agents disappear very quickly from the body. They are excreted within a few minutes, which means the imaging time you get is very inflexible. With SpagoPix, it's quite the opposite, actually. SpagoPix accumulates in the tumor and lightens the tumor, whereas the background remains unenlightened. This tumor selectivity can help reduce the number of false positives, we believe. It's also based on manganese, which is an essential element in our bodies. We get it with food and water every day, and there is a mechanism in the body to take care of excess manganese. So it's perceived to be less harmless than gadolinium. With the particles, we also can achieve a longer retention time in the body, allowing for more flexible imaging. So taken together, this makes SpagoPix a potential new agent to improve MRI, and especially MRI precision in solid tumors. Now, so how do we use this? We have chosen to focus initially on breast cancer. And the reason for that is that we have preclinical data that is solid and supports this. There's also strong support in the literature for use of these types of agents in breast cancer. And not the least, MRI is already an established technology in breast cancer. It's already in use for the characterization of these patients. And of course, breast cancer is a huge indication, as you all know. And a large fraction of patients already undergo MRI on an annual basis, especially in the US. There's also a medical opinion for the increased use of MRI in these patients. And that is because MRI is a really good method. It's better than mammography, it's better than ultrasound for spatial characterization and aggressiveness assessment and so on for breast cancer. So, and a premium, a specialized product in this field is considered to give a premium pricing as well. So with that, Spago Pix is targeting a significant market and has also, not to forget, the potential to expand into other solid tumors. So we believe this product can be of great use for the whole MRI field, actually. Now, just a few words about our second product, which is a bit earlier in development, but targets an even bigger market. TumorAd is based on the same technology. It's based on our unique nanoparticle platform. It uses a radioisotope, and it's given systemically and by passive targeting accumulates in solid tumors. That means we can treat solid tumors from the inside with a radioactive agent. And we know radioactivity works, right? It's been around for a long time. It's still one of the cornerstones of cancer therapy. The trick here is to get enough of it into the tumors while saving the rest of the body, of course. And this is exactly our speciality within Spago. Also, with the current radionuclide therapies, there is sometimes problems with the logistics because radioactivity needs to be handled, it needs to be coupled to the active carrier and so on. In our case, it's much easier. It's a matter of having particles in one tube, having the isotope in another tube, mix them together at the hospital, shake, and then you're done. So this is, sometimes people forget that the actual logistics at the clinic is really important if a product will succeed or not. There are some recent benchmarks which is very favorable in this field. Xofigo was approved for skeletal metastasis coming from prostate cancer in 2013. Its predicted peak sales is now $1.2 billion. And a similar product, Lutathera, which is based on an antibody targeted towards somatostatin and is used for a niche indication within neuroendocrine tumors, was recently approved and is now predicted peak sales of $2 billion. So we are targeting huge markets here. And again, TumorAd holds potential for broad application in solid tumors. Our particles accumulate in many different types of solid tumors, and especially aggressive ones, which means that we can actually target a growing therapeutic segments, and there is a need for new therapies, as you all know. So just to sum up, and this here is the setup right now at Uppsala University Hospital for our clinical trial with Spago Pix in breast cancer. The study was started just a few weeks ago, and we have the first patients in, and all looks good so far. With TumorAd, as I said, we are working on a lead candidate, and we are making a lot of progress there as well. We have allocated more resources over the last year to this project. So the next milestones will be first interim data from the trial with Spago Pix, but also declaration of the lead candidate in the TumorAd project. Please don't hesitate to contact me with questions, and thank you for your attention.